So we're going to continue with the lymphatics and immune system, looking at adaptive immunity, or the third line of defense. So sometimes things are going to get past the first and second line of defense. It's still not enough. And so at this point, when the third line kicks in, the immune system is going to recognize, attack, and destroy. But then it's also going to remember that pathogen that enters the body. And it does this through specialized cells and antibodies that will render the pathogen harmless. This is going to make it a lot stronger for the future. So unlike the first and second line of defense, this is going to differentiate among the pathogens. So for each type of pathogen, your immune system produces cells that are specific to that pathogen. This is what distinguishes adaptive immunity from your innate immunity, your specificity and memory. So an antibody is just a protein that's produced in response to an antigen. Antigens are macromolecules that are going to elicit the immune response. It literally means generates antibodies. So your most common antigens are proteins and polysaccharides. So this is showing the antigen coming into the body, things moving into a lymph node where it will encounter those cells, and so you can have your B and T cells going on making their specific proteins. So ways antigens can get in, they can be inhaled, such as proteins on cat hair, triggering asthma. It can be ingested, so you could swallow it, like shellfish protein that could trigger a reaction. It could be introduced beneath the skin, splinters, injected vaccines, puncture wounds. And then antigens can be generated within the cells of the body. So these could be proteins that are encoded by genes of viruses that infected a cell, or aberrant proteins that are encoded by mutant genes like you would see in cancer cells. So we have two types of adaptive immunity: cell-mediated and antibody-mediated. With cell-mediated, you have cytotoxic T cells that are your CD8 cells that will directly attack the invading antigen. These are effective against intracellular pathogens and some cancer cells or foreign tissue transplants. Antibody-mediated will involve the B cells forming plasma cells. These can make your antibodies or immunoglobulins. These work best against extracellular pathogens or in fluids outside of cells. They're going to rely on T helper cells. Both of them actually use the T helper cells, and the two types of immunity can work together. So both B and T cells will develop from pluripotent stem cells that originate in the red marrow. B cells finish their development in the red marrow. T cells will develop from pre-T cells that will migrate from the red marrow to the thymus. Your helper T cells, or CD4 T cells, are one of the examples of T cells, and then the other ones are your cytotoxic T cells, or CD8 positive T cells. So immunocompetence is the ability to carry out the adaptive immune response. It has to have the antigen receptor that is specific to the antigen. When you have clonal selection, you have the lymphocyte proliferate and differentiate. This is in response to a specific antigen. The clone is going to be the population of identical cells that will all recognize the same antigen as the original cell. So lymphocytes undergo clonal selection to produce your effector cells. So your active helper T cells, the active cytotoxic T cells, and the plasma cells. So these are going to die after the immune response, but it's a way for the immune system to say, I need help. I need more of me to do the job. And it actually gets more cells that are exactly like it to do the job. Memory cells don't participate in that initial immune response, but they're going to be there to respond to a second invasion that could happen in the future. And they'll proliferate and differentiate into more effector and memory cells. They have long lifespans. So this just shows here how you've got your primary lymphatic organs, the thymus and the red marrow, and the mature T cells that are going to come from the thymus, the mature B cells that come from the bone marrow. And so you can see how it will lead to your antibody-mediated immunity and your cell-mediated immunity. So antigens have two characteristics. They need to be immunogenic so they can provoke an immune response, and reactive, able to react with specific antibodies if it's provoked. So it may be the entire microbe, but usually it's just a small part of a large antigen will trigger the response. So we call that part the epitope or antigenic determinant. Haptens are low molecular weight antigens. They actually have to attach to another carrier molecule to be big enough. An example would be penicillin when it causes an immune response. So we have a huge diversity of human antigen receptors. They're able to bind at least 1 billion different epitopes. And this occurs because of genetic recombination that would involve shuffling and rearranging a few hundred versions of these several small gene segments. So the MHC, or major histocompatibility complex, or human lymphocyte antigen, the HLA, is important for this. It's important in organ transplants that you have enough similarity in these molecules to avoid severe organ rejection. It's generally not going to be an exact match, and exactly what percentage of a match you need is going to depend on the tissue type. So some things you can get away with about 50%, others you need much more. Bone marrow needs to be a really high match. So the normal function is to help the T cells recognize foreign or self. So class 1 MHC is built into all body cells except red blood cells, where the class 2 is only on the antigen presenting cells. So B cells can recognize and bind to antigens, but T cells have to have their antigen processed and then be presented to them. The antigenic proteins are broken down into peptide fragments, and that's what gets associated with the MHC. So you have when you have antigen presentation, you have the antigen MHC complex that gets inserted into the plasma membrane. And the pathway is going to depend on whether or not the antigen was from inside or outside the body cells. So exogenous antigens are present in the fluid outside the body cells. Your antigen presenting cell, or APC, which would include things like dendritic cells, macrophages, and the B cells, will ingest the antigen, and then they process it and put it next to the class 2 MHC molecule in the plasma membrane and present it to the T cells. With the endogenous antigens, they're inside the body cell, so the infected cell has to display the antigen next to class 1 MHC. So this is showing here the synthesis of your MHC molecules here and the process of displaying the antigen. And this is showing the process of displaying the antigen, this time for an endogenous antigen. So with cell-mediated immunity, we have to activate the T cells. So the first signal in T cell activation is a T cell receptor recognizing and binding to the specific foreign antigen fragments in that antigen MHC complex. So you have CD4 and CD8 proteins that are co-receptors. They need to have a second signal required in order to be activated. We know for co-stimulation there's 20 known substances that can do this. Cytokines, plasma membrane protein molecules are a couple of them. The idea is to prevent an immune response from occurring accidentally. With anergy, you've got recognition without co-stimulation. This can happen in both B and T cells, and this can lead to a prolonged state of inactivity. So when we look at activation and clonal selection of the helper T cells, most T cells are going to display the CD4, will develop into T helper cells. They recognize exogenous antigen fragments associated with the class 2 MHC on the surface of the APC. So after activation, they undergo clonal selection. It's going to make the active helper T cells and memory helper T cells. So these helper T cells will secrete a variety of cytokines. IL-2 is one example that's needed for almost all of the immune responses. So those memory helper T cells are not active, but they can quickly proliferate and differentiate if you get that same antigen back again. So they already know how to do the job in the future. So this is showing your antigen presenting cell, your antigen recognition, and your clonal selection, where you proliferate and differentiate the cells you need. And this is showing activation of the CD4 positive helper T cell. So you have your antigen presenting cell encountering the antigen. The T cell receptor on the surface of the CD4 is going to bind to the MHC antigen complex. The cytokines will cause the T helper cell to proliferate and differentiate. And this can produce your co-stimulatory molecules. So this again is showing here the antigen presenting cell, and then you can have it be extracellular, intracellular activation, and the different kinds of responses. So the activation and clonal selection of the cytotoxic T cells, most cells that display CD8 positive will become cytotoxic T cells. They target the cells that are self-carrying endogenous antigens. They recognize the antigen combined with the MHC1. So maximum activation is going to require presentation of the antigen with MHC2 to cause the helper T cells to produce IL-2. Then they undergo clonal selection, and these will attack body cells by releasing perforin and granulysin to cause cytolysis. Granzymes will trigger apoptosis, so it's sometimes referred to as the perforin granzyme pathway. Your memory cytotoxic T cells don't attack, but again, they're waiting for the antigen to reappear again. So this again shows your antigen recognition, co-stimulation, and creating your active cytotoxic T cells that can undergo your clonal selection and proliferate. And this is just another variation. So here you can see where you've got the granulysin perforin that's going to form a channel to allow the cell to be lysed, and then the granzymes triggering apoptosis. So your regulatory T cells are suppressor T cells. They have CD4 and CD25 on the surface, and they will suppress T cells against themselves. And here is showing your action of that regulatory T cell. So with the Th1 response, again going for your intracellular microbes. The Th2 for extracellular microbes, bacteria, and helminths. Helminths are your worms. So with antibody-mediated immunity, you have activation and clonal selection of the B cells. When they're activated, your antigen binds to the B cell receptor. They can respond to unprocessed antigens, but the response is much more intense when the B cell processes an antigen, takes it into the B cell, combines it with the MHC2, and moves it to the plasma membrane. Then the T helper cell can bind and deliver co-stimulation with IL-2 and other cytokines. So you'll undergo clonal selection, you'll have plasma cells start to secrete antibodies, and then you'll have memory B cells that won't secrete antibodies, but they're there for the future again. So this again is showing here where you can have the B cell receptor, you can have them become activated, and it's showing the comparison of activated in here, forming your B cell clones and your memory B cells. So antibodies, they can combine specifically with the epitope of the antigen that triggered their production. So they're going to be very specific to what they will bind. So they belong to a group of glycoproteins called the globulins. They're immunoglobulins. You've got 4 polypeptide chains, 2 heavy chains, 2 light chains. The hinge region, this is going to be, an antibody can be T-shaped or Y-shaped. And then you've got the variable regions at the tips of the H and L chains. So you have 2 antigen binding sites that are bivalent. The C region or constant region is the remainder of the H and L chains. And it's the same in each of the 5 classes that will determine the types of reactions. So here you've got your L chain, your hinge region, the H chain here, and then you've got your antigen binding sites. So your IgG antibodies, they're a monomer, they're 80% of the serum antibody. They're able to fix complement, found in the blood, lymph, and intestines. This one is able to cross the placenta. This allows it to enhance phagocytosis, neutralize toxins and viruses, and protect the fetus and newborn. The half-life is about 23 days. So it can be increased in old or chronic infections. Your IgM, it's a pentamer. So you've got 5 of them connected by these J chains. This is 5 to 10% of serum antibodies. They fix complement. They're in the blood, lymph, and on B cells. They will agglutinate the microbes first. So they're the first antibody produced in response to infection. They've got many more antigen binding sites exposed out here. So they would have increased levels when you've got a relatively early or new or acute infection. This is also the one that would do your ABO blood type reactions. It's got a short half-life, only 5 days. IgA, it's a dimer. It's joined by the J chain again. It's 10 to 15% of the serum antibodies. It's present in secretions and provides a lot of mucosal protection. Its half-life is relatively short, again, about 6 days. IgD, this one is a monomer. It's about 2% of the serum antibody. It's present in the blood, lymph, and on the B cells. It can initiate the immune response. Very short half-life, 3 days. IgE, monomer, it's only about 0.002% of the serum antibody. It's on the mast cells, basophils, and in the blood. This one's involved with allergic reactions as well as parasitic worm infections. Half-life, again, very short, 2 days. So this just gives some comparisons here. So antibody actions, they're going to neutralize the antigen, immobilize the bacteria. They can agglutinate and precipitate the antigen so they'll stick together. They'll enhance phagocytosis, activate complement. They can be a defensive system for over 30 proteins. So they'll destroy the microbes various ways. They can cause phagocytosis, cytolysis, inflammation. They can act in a cascade and have one reaction trigger another. They'll work with complement and activate C3 that will begin that complement cascade to lead to phagocytosis, cytolysis, and inflammation. So this is just showing different things that antibodies are able to do when they bind to antigens. And this is another illustration of things going on in the immune system. So immunological memory. You have thousands of memory cells after the initial encounter. So next time the antigen appears, you can proliferate and differentiate within hours. So it's a very quick response. One way we can measure the amount of memory is with an antibody titer, the amount of antibody in the serum. So your primary response is slower, the secondary response is faster and stronger. That's the idea with vaccination, is to hopefully have it be a secondary response when you would encounter the pathogen. So when we look at active immunity, this is the person who makes their own antibodies. Where passive, the person is going to receive antibodies from someone else. So passive is very temporary. This is comparing the immune responses here, where you look at IgM and IgG and their levels. So showing a primary response and secondary response of the levels. With natural immunity, it just happens. With artificial, it's caused by man. Most of the time, it's a needle involved with it. So natural active acquired immunity, the person would be something like coming down with measles, where artificial active acquired immunity, the person gets a vaccine. With artificial passive acquired immunity, the person is going to get someone else's serum with antibodies. So it would be something like a monoclonal antibody treatment or anti-venom if it's against a venom from like a snake. Natural passive acquired immunity. This is going to be a natural way of acquiring those artificial antibodies. So if somebody else has made the antibodies, it would be the baby receiving antibodies from the mother's milk with the colostrum. So again, this is just a table that compares those things. So recognition and self-tolerance are important here. Your T cells have to have self-recognition. They need to know what's your MHC. They also need to have self-tolerance. They need to not react to peptide fragments from your own proteins. So your pre-T cells in the thymus, they will develop self-recognition through positive selection. So the cells that can't recognize your own MHC undergo apoptosis. Self-tolerance occurs through negative selection, in which the B and T cells that recognize self-peptide fragments get eliminated. They can undergo deletion, where they undergo apoptosis, or anergy, where they'll remain alive but just become unresponsive. So when we look at some immune disorders, primary immunodeficiency means part of the immune system's missing or not functioning. There's over 400 types of these. These people are more likely to get severely ill. With a type 1 immediate hypersensitivity, it's IgE mediated. So your antigen and IgE bind to the mast cells and basophils. This would be things like your seasonal allergies or hay fever, local anaphylaxis, and hives. So it can also be a systemic anaphylaxis. Type 4 delayed type hypersensitivity is cell mediated. So you have the T cell antigen interactions that cause activation in cytokine secretion. It requires being sensitized, so that would respond 24 to 48 hours after exposure. There's a lot of these, again, poison oak or ivy, contact dermatitis, graft rejection, TB reaction when you get a TB test is actually looking for this. So allergies typically refer to that type 1 hypersensitivity response. The autoimmune diseases, the immune system mistakenly attacks healthy cells. There's a lot of conditions that are a result of this. None of them are curable at this point. Things like rheumatoid arthritis, lupus, celiac disease are just some examples. With HIV infection, the person has an infection with the human immunodeficiency virus, which is a retrovirus. It infects the CD4 positive T helper cells that affect co-stimulation. So when you have enough of those damaged, you're going to start to see the disease become a problem when you can't have the co-stimulation. AIDS, or acquired immunodeficiency syndrome, is that advanced stage of HIV. Here your immune system is badly damaged because it is not able to do that co-stimulation. So some treatments include antiretroviral therapy. We do have pre-exposure prophylaxis and post-exposure prophylaxis that's really helped to reduce transmission. However, it doesn't eliminate it altogether. So when we consider the diagnosis become AIDS, the number of CD4 cells falls below 200 cells per cubic milliliter of blood. In somebody that's got a healthy immune system, you have between 500 and 1600. The other thing that leads to a diagnosis of AIDS is developing one or more opportunistic infections, regardless of their CD4 count. So some infections you just don't see very often in a person that does not have AIDS. This is a nice little table here that compares the different hypersensitivities. So we talked about type 1 and type 4 in here. There is a type 2 and type 3 as well. That typically those are covered in microbiology. And that is the immune system.